This video describes step-by-step procedures on using biomembrane force probe, or BFP, to measure molecular spring constant.
Biomembrane force probe BFP has recently emerged as a native cell surface or in situ dynamic force spectroscopy nanotools. The main aim of this technique is to measure single molecule binding kinetics.
This data analysis procedure is a simple but powerful method to acquire biomechanical information, specifically the dynamic force and deformation of the cell membrane expressing molecules.
During the BFP experiment cycle, the probe micropipette with the red blood cell and probe bead, the target cell, and the ligand receptor molecular bonding between the probe and the target can be considered as a serially connected spring system. Based on Hooke's law, the reciprocal of the total spring constant of a serially connected spring system is equal to the sum of the inverse spring constant of each single component, as the equation shows. The spring constant of red blood cell is calculated based on Evans model, which is dependent to the radii of the probe micropipette inner orifice. The red blood cell, the circular contact area between the red blood cell and the probe bead, and the micropipette aspirating pressure. The BFP raw data is obtained by a homemade LabVIEW virtual instrument, which controls the movement of the target micropipette by a piezo translator. A full BFP